Hello everyone. So today I'm going to show a demo on the procedure to plot a PCA biplot with its genotype name or any specific treatment names. So usually in RStudio, the PCA analysis or the biplot analysis gives you some default numbers for your genotype values or your treatment coordinate values. So today we are going to try to label any name we are going to assign a name to those genotypes. So we have a lot of names for our genotypes in plant breeding, right? So we are going to try to incorporate those names into our biplot. So for that, first of all, we need the RStudio. So I hope everyone are familiar with the RStudio. So we need to open that one and import our data set first. So for that, into your right hand side, you can see an option called import dataset. Just click on there and click on from Excel. Go on to browse your file. So I have saved my file under the name PCA data. So this is my file name. Just click on it. So here you can see that the first column is my genotype along with its name. Either you can give the name or any numbers, any specific numbers or specific codes, whatever you want, you can incorporate it there, which is accompanied with all the necessary traits. I have around 1, 2, 3, so around 9 traits. So whatever the number is doesn't matter. I have taken the mean value. Usually for PCA we only go with the mean value. So it's been loaded. So click on this import, switch down to the right hand side, the lower right hand side, just click import. So you can see the PCA data here. So once the PCA data is in your console, now you need to write down some codes. It's a pretty simple one. I have already done a video on how to do the PCA biplot analysis using the factor minor library and the factor extra library. So here also we'll be using those two and 2 additional libraries. So total we are going to use 4 libraries. So the first library is library ggplot 2. So we'll type that, control enter to load that, and another library is factor minor, and the third one is library factor extra. So load that too, and the last library that we might need is library ggrepel. So these are the 4 libraries that we are going to use today. So make sure that file name is pretty much simple. I have given PCA data, all are case sensitive, so make sure you type in that very clearly. So now we need to find the PCA value. For that, we'll be using the function PCA. So you can see the RStudio gives you the hints or the suggestions where I need this PCA function from the factor minor library. So I'm going to select that. So, and once you select that, you now need to specify the file name. The file name is PCA data. So click on that. And once you selected that, we have as first column is about our genotype name, which is a character, and other 9 columns followed by that genotype column is our data, which is the numerical data. So we need to specify that column. To specify that, we'll be opening a square bracket, and inside which we need to specify the columns that is containing all our trait values or the numerical values. We need to specify this here. So just check what are the column numbers. So here just hover around that number. You can see that my first trait is in column 2 and my last column is in column 10. So I need to specify like 2 colon up to 10. So it will take the data from 2 to 10. Then just type in the function graph, which is false here. So once you do that, close the bracket and run the code, and you will have all the types of PCA values stored. So we can save this PCA scores in a string or a name. You can give it a name for that. Like I am going to give it as my PCA with an underscore. Just I am going to store those values inside this so that it will be easy for the downstream analysis. So just copy that, paste it here and set it. So it's now the PCA data for my numerical values have been stored under the name my underscore PCA. So you can always view it, my PCA. So you can see that it's already there. So once you click that, you can see there are a lot of values that has been stored under different cells, like aging vector values, the variance values of var means the traits coordinate values, its contribution values, the individual genotypes value, everything is saved under different cells. So we need these separate cells for our further analysis. So once we are ready with the PCA values, we need to specify the genotype column and store it under a different name so that we can label it when we plot the graph. So for that, what we'll be doing is that we'll create another name called, say, geno underscore name. So you can name it as per your wish. I'm just giving it as geno name is equal to. Now I need to specify the data file, which is PCA data. Click on that and click on the $sign. Then you can see all the columns pops up. So we need to specify the genotype name, which is under the name geno. So the column starting from the name geno has all the data that is necessary for labeling my biplot. So I'm going to click on that. Now press Ctrl Enter. So that has saved that particular geno column under the name geno by name. So now we will be going into the plotting of the biplots. So for that, we need the function fi. Is a definite. When you just type fviz underscore PCA, you can see various functions of the library. Facto extra. Here we are interested in fviz underscore PCA underscore biplot. Once you click that, we are now ready to plot the graph. So again, we need to specify the PCA data, which was saved under the name my PCA. So I have specified that. Now I need to specify the type of the graph, like how the points I need to be, whether it needs to be square, points, something like that. So for that, we'll be using the function geom. So, and under geom, we are going to use point, comma. You can change those things. It doesn't need to be every time point. You can just search what are the other options that we can get, like to get this will give a points biplots. The biplots will be in point structure. You can have triangles, squares, anything you like. Now we need to specify the axis. So axis, these are default things like 1 to 2, like the x and y axis. Once that is done, we need the colors. So for that, col.var for the variables, that I would be going with some black color, comma, and we need to specify a theme. So for that, I'll be going with GG theme. So the GG theme is equal to theme underscore minimal. So this is also a function of ggplot2. They have some default functions which determines the theme of your biplot. There is theme classic, theme gray, theme minimal, and few others, I think some 8 themes are there. I'm going with the theme minimal. And once you have that, just close your brackets. So this will give you a pretty much simple biplot, your usual one, but we need to label the genotype name. So for that, we need to specify some other things, some extra things. For that, give it a space and just give a plus symbol and give it an enter. And now specify your another function called geom underscore text underscore repel, which is a function of ggRepel library. Just click on that. And here also now we need to specify some data frame from the PCA values, and we will be labeling our biplot here. So for that, type data equal to data frame. So that's the base function. Click on data frame and now specify your data frame that you need in your biplots. For that, again, my necessary values are stored under the name my underscore PCA. So under that, give a dollar symbol so that you can see what are the things we need. So we need the individual, that's the genotypes value, so which is under the IND, which is the individuals. Just click on that and again give a dollar sign, so which will give you 3 more functions, the datas like coordinate datas, the contribution datas and all. So we need the coordinate datas for the PCA coordinate datas. So for that, you need to choose the coord. Once you choose that, give it a comma and specify the genotype names for that. So for that, we have saved all the genotypes name under geno name. So give it that. So the genotype name should be specified there. Now close the bracket, give it a comma there. And I'm going to click and enter. So if you want, you can just type it in one line. Just to avoid confusion, I'm just giving some indentations in and there. Now I need to specify the X and Y axis. For that, I'll be using the aesthetic functions of the ggplot2, and I'm going to give X axis a value. So X equal to, so let's say dimension 1. You can also put like PCA1, PCA2 axis, something like that. I'm giving dimension 1, comma Y equal to say dimension 2. Now I need to label those points. So again, I am going to specify what I need to label it. It will be again the geno name. Yep. Now again, close the bracket, give it a comma there. And I'm going to just specify the dot sizes. You can change it. These are some few default things. And the color that I'm going to prefer is red. Again, close the bracket. Now run the and it's still running. So you can see the plot here. So it might take some time according to how big your data is, but finally you should get your data. So you can see that every point, genotypes are in some black, very dark black points. And you can see the trait coordinates also, how it is correlated. And interestingly, all the points has a name, like all the genotype names are here. You can mix it up with numbers, names, codes, anything you want. Even if you want only few 5 or 6 genotypes names to be included, you can do that. And remaining you can just specify some simple numbers so that you can easily see them. So that will be it. So I'll be giving this code in a notepad. I'll be giving this sending this codes into a notepad, and I will be posting it on our website geekyresearcher.com. So that's it for